We will move to our next speaker. It's Professor Bastiaan IBELINGS from the University of Geneva. Bastiaan is head of the Laboratory for Microbial Ecology, and he has a strong focus on phytoplankton in lakes and their grazer. And he tackled biodiversity at different levels, the evolutionary origin, but also the population level and the community level. And finally, the role of the biodiversity in the provision of lake ecosystem services.
Like this. Is it on? Yeah. Some say that the first paper ever written in ecology is The Lake as a Microcosm by Stephen Forbes. So, in that paper, he says if you're interested in a certain fish species in the lake, you won't get very far if you don't study the interactions of that species with other species as well as with the environment. So there you have it, ecology for the first time in a paper. Why do I like the lake as a microcosm? I'm a lake ecologist. I could not imagine having to work on oceans. A lake, nicely defined boundaries. You know how much carbon, nitrogen, phosphorus comes in. So it's a good model system to study the complexity of an ecosystem. So here's a picture from a Swiss lake. And although I like when lakes look like this, most people do not. These are blooms of toxic cyanobacteria. And I'm going to talk to you about the threats to lakes, the functioning of lakes, and therefore the services of lakes and what we can do about it. And let's begin with this purple cyanobacterium that has the name Planctotrix rubescens. So this is the kind of environment that Planctotrix likes. It likes its life calm, stable, not a worry on its mind. And there is some music attached to it, because I thought instead of describing the habitat of Planctotrix, I'll play some music that nicely describes the kind of conditions it lives in. Because you're not going to remember anything I say to you, but you might remember the music. That's the poor fate of us scientists, that we don't touch people's hearts usually. So finally, it's the arts and not the sciences that really matter in our societies. But I don't think the music was transferred, when I sent the file. So let me see if I can connect to Spotify and play the music for you. Can you hear that? But it doesn't pick. You get the idea, right? Anyone know which music I just played? No? That was Bach. And Air by Bach. Why air? Because the key trait of this cyanobacterium, it has gas vacuoles in its cells, and it can float because there's air inside the gas vacuole inside the cell. So that's, a picture of the life of Planctotrix in its habitat. But now this, you can see where Planctotrix lives, in this nice, calm, stable layer at the top. But look what's underneath it. Turbulence. So I'll play you another song that shows the risk Planctotrix faces. Do you know which song this is? Bach again? Yeah, his late works. No, that was Metallica, Master of Puppets. So why do I play Master of Puppets? So once it sinks into this zone of turbulence, it's no longer in control where it sits in the water column. So in a stable environment, more like Bach, it uses its buoyancy, its gas vacuoles, to position itself where it wants to be in the water column of the lake. In a turbulent environment, it's the puppet master. It's turbulence that determines where you are as a plankton cell. No control. Now, this picture here is the Battle of Lac de Murat, I think 1476 in Switzerland, where the soldiers from Burgundy were slaughtered by the Swiss. And that colored the lake red. So this is not Bœuf Bourguignon, but more like Sang Bourguignon. And that's where the name of Planktontrix comes from. So in German, it's Burgunder Blutalge, the blood of the Burgundy soldiers that turned the lake red. And below you can see what it looks like. It's a filamentous cyanobacterium. So here's a study from a PhD student that defended earlier this week, Ena Suarez. And I use this to say that even in a lake that we use as a model system to study ecology, that we think is well-defined, there's often surprises. And here's a surprise with Planktontrix. So where does Planktontrix live? If you know what a deep lake like Lake Geneva looks like in summer, it's stratified. So there's the Epilimnion at the top, there's the big part, the Hyperlimnion, deep in the lake, and there's a transition zone in the middle, the Metalimnion. And that's where you have a very steep temperature gradient, and that's because the temperature gradient gives stability, and that's the Bach music is provided by this temperature gradient. So you can see here that this is a history of eutrophication of Hallwyl, another Swiss lake. So we start here in the 1980s with very high phosphorus levels, and then they started to clean up the lake. And you can see initially, actually, the red line, Planktontrix wasn't there. Why not? Because it lives in this deep layer. It forms a deep chlorophyll maximum in the Metalimnion, in this thermocline region. And in a eutrophic lake, there's simply no light that travels down to depth because it's all taken at the surface. So first surprise, they started to clean up the lake, reducing phosphorus at huge costs. And what did they get? A bloom of toxic cyanobacteria. Look at the red line. Goes up to much higher levels than in the dirty days. So bad surprise, right? But they continued to clean up the lake and it went down. You can see the same here in those turbidity profiles. High biomass in the early 2000s and also quite close to the lake surface. And then with ongoing re-oligotrophication, so the opposite of eutrophication, bringing nutrients down, re-oligotrophication, Planktontrix not only went down in biomass, not a big surprise, but it also went deeper, and that is a surprise. And again, this goes back to the difference between Bach and Metallica. See where it was sitting early on. The red line is Planktontrix. This is the depth of the lake. And the purple line is the depth of maximum stability. So that's a physical number that explains us where the lake is most stable. And it's in this thermocline region where we have the density gradient. So, and that's where Planktontrix has evolved to nicely sit in those stable conditions. And it did early on. In its heydays, it exactly matches the depth of the maximum density gradient where it's stable. But then for some reason, it starts to sink down. And it starts to sink down well below this stable layer. It even now has left the Metalimnion, this intermediate zone, the blue line here. It's now a hyperlimnetic cyanobacterium. And that was another surprise that we did not foresee. This is now really getting into Metallica. Metallica is in the blue. So early on, it's sitting nice in the stable red layer of the lake. And it's been sinking, and it's getting very close to the blue turbulent layer at depth. So there's turbulence at the top from the wind, there's turbulence from internal waves near the sediment of the lake. And if it sinks a little bit deeper, it can no longer stratify, it cannot form its deep chlorophyll maximum, it might disappear. So why is it sinking? I told you about the gas vacuoles, so they have gas, air, inside a protein structure that provides buoyancy. They can float. But if they're photosynthesized, they build sugars, and sugars are dense. So at some point, the sugars overtake the lift from the air, and they start to sink. When the lake was cleaned up, the top part of the lake became very clear. More light penetrates into the lake, and at some point, they build up so much sugars that they start to sink until they reach a deeper layer, a darker layer, where, again, they can stratify, where that buoyancy is neutral, where they have the same density as water. So they don't want to go down, they're forced to go down by their own key trait, which is buoyancy. Also, by going down, initially again, you can see this is where it would sit early on. It just has enough light from the surface, but also where the phosphorus is in a deep lake is in the hypolimnion. So the ideal position to be, light from above, nutrients from below, and that's the typical Planktotrix niche. But now with ongoing oligotrophication, actually, even deep down in the lake, you can see how we go from blue to yellow. There's very little phosphorus. So the whole typical habitat is disappearing. So here's like a take-home message of this little story. Planktotrix, the dominant toxic cyanobacterium in deep peri-alpine lakes, may have changed its habitat and became a hypolimnetic bloom-forming species. Drivers of environmental change, re-oligotrophication and climate change, combined with buoyancy behavior of the cyanobacterium, may explain this. There are real consequences for drinking water production. You know, one key service that we get from lakes is drinking water. It's the largest reservoir of fresh water is in lakes. Lake Geneva, it's more than 1 million people that rely on Lake Geneva for their drinking water. You would extract drinking water in a lake from depth, also to avoid, the blooms of toxic cyanobacteria in the metalimnion. Now that it's sinking into the hypolimnion, as a drinking water company, you need to rethink your strategy of where to extract your drinking water. So that was one message, expect the unexpected. And here's another one. Another thing that we did not foresee in Lake Geneva, in 2021, the whole lake turned brown. So we're used to nice, transparent, clear, clean lake, and all of a sudden it looked like this. This is Uroglena, a chrysophyte. It's not toxic, but it smells bad. So, people start to call and make complaints. And of course, then they ask the scientists what happened. So these are two extreme events in a row. A very wet August month in 2021, bringing a lot of nutrients, but also carbon from the lake watershed into the water. This is a mixotrophic algae. It can use carbon. It can feed on bacteria that feed on the carbon coming into the lake. And the very wet August was followed by a very warm, stable September month, and they can swim. So two key traits that led this species to bloom for the very first time, linked to two extreme events linked to climate change. So Chris also just talked about predicting plankton, and predicting is hard, in particular if it's in the future. So here's a paper from Medul Thomas, maître assistant in my team in Geneva, and he looks at the predictability of lake plankton over timescales of hours to years. And you can see how predictability, this is the R squared between observations and predictions. Over days to weeks, it's fairly okay. But if you go longer, months to years, it starts to break down. The other thing we show here is that if you have high-frequency data, you do better. Your prediction goes up, the quality of prediction goes up with high-frequency plankton data. So, limnology, the study of lakes, was invented by Alphonse Forel, who was a local from Lake Geneva. He lived in Morges, and he wrote a book, Le Léman, une monographie limnologique, and in that book, he writes, I propose to call this study limnology, the oceanography of lakes. So that's the first time limnology was used by Alphonse Forel. You can see him sampling Lake Geneva in the 19th century, and this is how we still do it, limnology. So that's why I say lakes in a changing world. We're facing 21st century environmental challenges with 19th century techniques to collect our information. So there we have a challenge. So classical limnology, and it's the same for Lake Geneva, so this would be somewhere here is the center of the lake, where twice per month people go out and take samples, also take samples to put under the microscope and count your phytoplankton. They've been doing this for more than 60 years, so extremely variable to see and understand how the lake changes. But there's, the problem that if you think of phytoplankton, they're microbes, so they're very short generation times. So basically, if you go once every 2 weeks, there's a lot that has happened in the meantime that you don't understand. And that's something we describe here in this paper that led us, I was in Zurich at the time at ERWAG, to build a first automated platform for phytoplankton monitoring. The problem with going infrequently, twice per month, is shown here. If you go twice per month, you're basically getting the red dots, right? So you could conclude that phytoplankton dynamics, or imagine this is primary production, it's quite stable over the whole period. And in fact, what you're missing is all the ups and downs. So this is the Nyquist theorem. So basically it says you need to sample twice as frequent as the phenomenon of interest to be able to capture, the dynamics and to be able to understand what is happening. And here's a good example going back to Planktotrix, and this time in Lake Zurich. So for instance, look here at the cell density in panel A. So these are two classic samplings, 2 weeks apart. And again, you would conclude there's no dynamics. Then if you do, sensors that pick up the pigments of this red cyanobacterium, you can see there's a whole bunch of things happening in between your two traditional sampling points. And that's, again, why we need to change things, how we monitor plankton, because otherwise we'll never understand the true dynamics and the true impacts environmental change are having on plankton that are feeding the rest of the food web. So here's Lake Geneva, and probably we can say that it's the best studied lake in the world. I mean, look at all the institutes that are around the lake. So this is us here at the end, Petit Lac. Then there's in Lausanne even 2 universities, UNIL, and the Federal University, EPFL. There's the Federal Research Institute, ERWAG, that does a lot of work on the lake. And on the French side, in Thonon-les-Bains, we have INRAE. So a huge number of scientists all working on Lake Geneva, but until not so long ago, basically everybody did their own thing. So one thing, we wanted to have a floating laboratory with loads and lots of, dozens of different sensors to do high-frequency measurements. But the other aim I had from the beginning is to bring all those people together by giving them access to our floating laboratory. So this is what we call l'Explore, for Le Mans Exploration. And you can see it's a 100 square meter floating laboratory in Lake Geneva. And again, it's accessible for everybody that wants to do science at any given time. There'd be about 30 different scientific projects ongoing on l'Explore, bringing disciplines, very different disciplines together to study the lake in an automated, in a high-frequency way. And here you can see, a paper where we describe this. This is called the TETIS profiler, which is hyperspectral measurements over the depth of the lake. Again, it's programmed to do this, I think, every 3 hours. There's a profile. And this is critical because it's the same type of information that you pick up with satellites. So we often get the question, okay, very nice that you have l'Explore, but it's one spot, one location in the lake. What about the rest of the lake? Well, with those kind of data from l'Explore, we can improve the algorithms for the satellite images, like the Sentinel that was shown yesterday, and that way we can also capture, high spatial resolution in addition to high temporal resolution. What about phytoplankton? So phytoplankton, unless you have dozens of PhD students in your lab, taking samples at the required frequency is actually several times per day. Looking at Nyquist, that's what you need to do. That is impossible with traditional means. The number of microscopic samples just explodes. So we have this instrument on the platform, which is a flow cytometer specifically designed to do phytoplankton. because it can do from the very large to the very small, all in one machine. Orders of magnitude difference in size. But we also have flow cams for imaging. So it takes, it's programmed, it takes profiles every 3 hours, 1 meter intervals. Each depth, it would count 60,000 phytoplankton cells. And that's how we hope to better understand, the true dynamics and the true spatial distribution of the plankton. Here's some of the scatter plots you get. So this is 2 types of fluorescence. There are all sorts of parameters you can use and plot to try to tease apart what you find. So here's inside the cabin, there's what we call moon pools. So there's access to the water, and this is just the big machine with the cable that is programmed to bring the machine up and down, to bring the samples up to the flow cytometer standing there. And for instance, this cluster here, the blue cluster, are cyanobacteria in the lake. It does take images, but poor quality images. The images are important. That's why we now have upgraded to better quality images to try to improve this process of automated plankton annotation using L'Explore. Right, so in order to manage changing lakes and maintain lake services, we have to improve our observations. If we do not observe properly, that is, at the correct temporal and spatial scales, we do not know if and how lakes are losing their capacity to adapt to environmental change. Right, and here's one of those satellite Sentinel-3 images during the Euroclina bloom in the lake. You can nicely see the mesoscale eddies that are quite typical of those large lakes. Right, so I've been talking about nutrients, eutrophication, causing blooms of toxic cyanobacteria, re-oligotrophication, cleaning it up, getting nasty surprises when they actually get worse rather than better. But of course, in a lake, there's plenty of other stresses, and we've talked about them those days. We have effects of climate change, we have invasive species, we have plastics, we have all sorts of things. And of course, in the end, if you're a Daphnia that lives in a lake, you don't get to choose. You don't get to say, today I only care about the plastics. I forget about the toxins coming from the cyanobacteria, or I forget that it's getting too warm. No, as an organism, you need to deal with all of that together. So the other challenge for our research is to bring it all together, to understand interactions. So here's one organism that is recent, but that is probably, in a paper we say this is the biggest change to occur to Lake Geneva since eutrophication of the 1970s. And here's the paper, An Abundant Future for Quagga Mussels in Deep European Lakes. What we've done is we've compared quaggas in the US Great Lakes, where they've been around for 30 years or more, with some of the Swiss lakes, where they have been around for 5 years. So we use comparisons between US and Swiss lakes to predict, and again, that's full of pitfalls, to predict where quaggas may go in future. And of course, you need to select comparisons: lake size, lake depth, lake morphometry, lake trophic status, the sediment type, the availability of substrate. They all make a difference for the quaggas. So we try to make comparisons that are as close as possible. Like for Lake Geneva, Lake Erie, the eastern part of Lake Erie is the best comparison for Lake Geneva, we found in this study. And you can see, if you look at this graph here, so the dots is where this is the year since quagga detection, and this is the quagga biomass in the lake. And the dots is where the lakes are now. So green is where Lake Geneva is now. This is where the US Great Lakes are after 30 years. So we predict that it's going to increase 9 to 22-fold even compared to today. Not only will they increase in numbers, also they will increase in size. Again, making the comparison shown here in this graph, and they will continue to go deeper. 5 years after the first mussel was found in Lake Geneva, if you now look at video images, there's not a square centimeter of sediment that doesn't have a mussel. Down to 250 meters deep. Below that, there are no mussels because there's no oxygen. but they're already everywhere. And if this is correct, it's going to go much further up. And we talk about lake benthification. Those mussels are so numerous and so effective in filtering the phytoplankton that they're stealing all the food from the classic pelagic food web. So instead of having Daphnia and Corrigon, we get benthic communities, benthic organisms, including benthic fish, that may benefit from this transition. So there's also a message there for the fishers. You know, you need to change. Now they catch Fera and Perch. They need to change, think about, you know, catching different species in the coming years. Right, I'm gonna maybe skip this. I never plan, you know, I just have enough slides to keep me going, but looking at the time, maybe I was gonna talk about tipping points. Let's go to the end. Maybe this is, how do I go up again? Can I? Can I go up somehow? The bottom? Okay. Just one message here about climate change, because now we've talked about nutrients, invasive species. Clearly, this is another big one. The big impact of climate change on those deep peri-alpine lakes is that the mixing changes in the lake. So Lake Geneva, we've not had a fully mixed lake for the last 13 years. So what you need for lake mixing, lake overturn in winter is both low air temperatures to bring the water down to 4 degrees, which is the most dense water that will then sink like a brick and mix the lake, and you need strong continuous wind from the northwest. And those conditions don't happen that much anymore, or not strong enough. So the lake doesn't mix completely, leading to a lack of oxygen in the deepest part of the lake. So the bottom 50 meters are back to becoming almost anoxic, like they were during eutrophication. In this case, it's not the nutrients, it's the climate change causing anoxia. And of course, we know with anoxia, the redox status of the sediment changes. There's a huge amount of phosphorus stored there from the eutrophic years. With anoxia, the phosphorus will be released, and that means that there's a risk of internal eutrophication, you know, a phosphorus bomb going off in those lakes. And here's a paper. I was going to talk about tipping points. Classic tipping points are studies of shallow lakes with eutrophication. Macrophytes disappear. Within a few months, lakes go from clean water, high biodiversity, to turbid water, loss of biodiversity, just blooms of toxic sand bacteria, not much else. In deep lakes, this is work of a PhD student a couple of years ago. Basically, the message is, without going into detail, if you have incomplete mixing because of climate warming, shown here, anoxia develops near the sediment, solutes, including phosphorus, are being released, and with that, with the solutes in the deepest part of the lake, the density of the hyperlineant goes up. So now we get a risk of a lake. It's already gone from holomictic, meaning fully mixed, to oligomictic. It doesn't mix completely. There's a risk with the solutes of it becoming mirromictic, so stratification based on difference in salinity. And that will be a lake very difficult to mix at all. Right, so let's skip these then. Lakes like Lake Geneva are under pressure from multiple drivers of change. Combined, these drivers, like eutrophication or eutrophication, climate change, and invasive species, are drastically changing the structure and functioning of lakes. Irreversible tipping points should be avoided to maintain lake services upon which we all depend. And this is then the last slide, and going back to what was said yesterday, yes, we're scientists. We study lakes, we've done so for decades, if not longer. That's very useful, but there's another type of knowledge. It'd be hard to look for indigenous Swiss people for Lake Geneva, like we can do like in Australia, for instance. They have a type of understanding, a different type of observations of ecosystems that are extremely valuable to match with our scientific approaches. In Lake Geneva, in this project Co-Fish, we start to work intensively with the fishers of Lake Geneva. So there's quite a few professional fishers. Then there's hundreds and hundreds of amateur fishers. They depend on the lake also for another type of ecosystem service, which is fish. And what I find interesting about co-fish, the proposal we wrote for the National Science Foundation, we did not have any scientific questions in that proposal, because we said the questions need to come from our interactions with the fishes of the lake. So we had a whole series of workshops over many months where we would, in the beginning, this is awkward, right? They said you're the first professor from all of those universities that really comes and talks with us, sits down and listens to us, and it's extremely interesting. So they developed the questions for the project that we have investigated together, more like traditional community citizen science, because they have dozens of boats, they can go out and sample spatially in a way that we cannot. But the important thing is the co-creation, you know. Science can be developed together with lake users, and that gives a whole different understanding, but it also creates trust. So now we have a level of trust between us and them that is important, I think, to manage the lake in the future. And this is CIPEL here. CIPEL is the Comité International Protection des Eaux du Léman, so it's Franco-Swiss. And also they now, they interact with our scientists. I'm on their scientific board. They also now more and more see the value of reaching out to the whole community. And I think that's important to maintain functional lakes, lakes that provide services, even under all the different stresses that we humans put on those ecosystems. And I thank you.
Thank you, Bastiaan. Trust of the society, yeah, that's the point. Questions?
Thank you for your nice presentation. You show that the external phosphorus in the lake decreased during the year, and maybe the euphotic zone was decreasing also. And I was wondering if the fact that Planctotrix go down near the metalimnion edge was not also to have the new phosphorus from the microbial loops in the aphotic zone.
No, that's a good point. So initially we could see that indeed by moving deeper, they were able to continue to access the phosphorus, which was going down even in the hyperlimnion, but they would move closer to that and maintain access to phosphorus. Now phosphorus in the whole water column is so low that that's not really a strategy. So what we see is what Planctotrix is doing is what we call oversummering. So the growth is in winter, because with the winter mixing, there's a last bit of phosphorus that is brought up to where the light is, the euphotic zone. So they grow in winter. The deep chlorophyll maximum that they form in summer, there's no growth. They simply maintain the biomass to wait for the better conditions that arrive in fall, in early winter, because that's when the phosphorus is still a little bit available. So instead of overwintering, because of effects of climate change, we see oversummering.
Thank you for your talk. You spoke about the Léman Lake. There is other lake around, like Annecy, Le Bourget. So is there a big difference in diversity in, let's say, phytoplankton in this different lake, and how is it the impact of climate change on these different ecosystems?
Yeah, so the colleagues in Thonon-les-Bains, they're responsible to follow three lakes, like the three lakes you mentioned, Le Léman, Lac Bourget, and Lac Annecy. They have very different histories. Like Bourget has a history of strong eutrophication and strong re-oligotrophication. Annecy has always remained oligotrophic, and Geneva is somewhere in between those two. Yes, so phosphorus went up to about 90 micrograms per liter, came down to, now we're back to about 10, 15 micrograms per liter, which is the long-term goal. But because of the difference in history, like Bourget had also a phase with strong development of Planctotrix, I don't think Annecy's ever had that to the same extent. And Geneva actually, it's not so much Planktothrix that is causing issues. It's other, not even cyanobacteria, like this Mugosia, which is a green alga that can be so abundant at certain depths that it's clogging the nets of the fishers. And while talking about phosphorus and fishers, of course, the first thing that comes up if you talk with, in particular, the professional fishers, they say you're making the lake too clean. You know, we need phosphorus in our system to produce plankton that feeds the fish that we can catch to have a living. And you can, you know, not take that seriously, but I think it is a serious question. You know, different lake services demand different levels of productivity for drinking water, for recreation. You know, clear water is fine. You know, it could be tap water. But for other users, phosphorus is not toxic. You know, it's a nutrient. So that's not only in freshwater, in marine systems, it's the same. You know, what is an optimal level of phosphorus to maintain? In the end, also like birds, bird numbers, they also depend on lower trophic levels to provide them food. So it's not only for fishes. In the end, I know biologists that are also saying the same. You know, we should stop cleaning up our lakes because who's going to feed them? And I'm not, you know, it's a whole discussion. Yeah.
I have another question about the mussel you spoke about. I was wondering, where is it coming from, actually? Because it's not under Genesis, right?
No. So they're actually there too, right? In the genus Dreissena, there's the zebra mussel that has been around for many, many years. Like in the Netherlands, for instance, it came in the 19th century. But the quagga is recent in Western Europe. It comes from the Dnieper, so say Ukraine. And as you know, maybe we've built a canal between the Danube and the Rhine, and that's like a superhighway for invasive species. So it's been a whole series of one species after the other making its way from Eastern Europe to Western Europe, and some of them damaging, others not so damaging. But the quaggas, I think, is one of those that will have a massive impact on lakes. And once they're there, there's not much you can do. There are some predators, some fishes, diving ducks. But the ducks, you know, they can only go 6 meters deep. If they need to dive deeper, the energy they get in return from feeding on the mussels is less than the energy they need to invest to dive there. So in a deep lake like Lake Geneva, they can keep the littoral zone somewhat clean, but the rest of the 300 meters lake, it's difficult. So the best thing is to prevent the mussels from spreading. So most lakes in Switzerland, France, I'm not so sure, don't have them yet. It's often boats like regattas that go from one lake to the other, where the mussel larvae, for instance, are transferred. So that is, just asking that people clean boats, paddle boards, and so on before they go to the other lake. Yeah.
Other questions? Thank you very much, Bastiaan.